Hello everyone. I'm making this video for the Thursdays and Fridays sections, but everyone can look at it obviously. There's your comic. It says, "Ooh, goldfish everyone, goldfish." So you can't see it, but the guy that's standing over top of the moat is pointing down at the moat being like, "Ooh, goldfish everyone." Anyways, the band you should check out is Spoon. I would play them, but then you probably wouldn't hear me for the rest of the presentation. Picture of fire. Your tip of the week. Men are not better than females. This is because there was an interesting TED Talk that Mr. Houston read, and also I just know that this is a mentality that some people have in this country and in grade 9, and I just thought I would say that because it's true, and if you have any questions about that, please let me know, and I will be more than happy to explain why this is true. Moving on. Alright, so we talked about cells. We talked about your nucleus. Your nucleus has your DNA in it. Your DNA is deoxyribonucleic acid. It's a double helix, and it holds all of the information that makes you you. So it's the blueprint of your cell. And the code of your DNA is A, C, T, and G. And then you make a copy of your DNA so that you can get that information outside of your nucleus to your ribosomes specifically, and that is mRNA, which is RNA, which is a single-stranded genetic code, and instead of T's you have U, so it's AUGC. And then with mRNA, it goes to your ribosomes. So what happens is it leaves your nucleus and it goes through your rough endoplasmic reticulum to your ribosomes. In your ribosomes, it goes through the active site, which is that yellowy part to this picture, in between the small and large subunit. And what it does there is the ribosome reads your mRNA and adds amino acids to it for every 3 letters. So if you have AUG, it adds an amino acid, and then you would have UUG or whatever, and then it adds a new amino acid, and it keeps adding 1 amino acid for every 3 letters, and it will glue all of the amino acids together. So you have a string of amino acids bonded together in the end result after your ribosome has read the whole mRNA, and then this string of amino acids is protein. The proteins get sent through vesicles, which is just a membrane-bound package. So it buds off of your rough ER, and it goes to your Golgi apparatus or Golgi complex. And then what happens is your Golgi apparatus tweaks a couple of things and then sends it off in more vesicles to wherever you need it in the cell. You can also find a very good diagram of this on page 200 in your textbook. It shows the whole process instead of just like a couple of pictures of the organelles. Now, always stay hydrated. And this is your mitochondria. Your mitochondria is the powerhouse of the cell. So what it does is it takes your glucose that you eat and it converts it into ATP, which is adenosine triphosphate. So it's adenosine, which is a chemical, and 3 phosphate groups on the outside of it. So what happens is you break the last phosphate off of ATP, so you get ADP, or adenosine diphosphate. So what's left is you have a phosphate that's usually connected to a protein or whatever it is that is using the ATP. And then that protein can then do something with the changing and breaking of the bond. Hi. So what happens is you make ATP, and then you use ATP for energy, and then after it's used, you make ADP, or adenosine diphosphate, because you broke one of the phosphates off of it, and then you use more glucose to make more ATP by just re-gluing the phosphate group back onto the ADP to get ATP again. If you have questions about that, just come and see me. It's easy to draw it on the board. Actually, I'll go back. One more time. Your mitochondria are really cool because they're believed to have been bacteria back in the day that a cell just ate. This is because there's two membranes around your mitochondria. Your mitochondria have its own DNA and they reproduce by themselves, so you don't actually make your mitochondria. And you got your mitochondria from your mom. These are chloroplasts. Chloroplasts are filled with chlorophyll A. Chlorophyll A is a green pigment. That's why plants are green. And what chlorophyll does is it takes the UV light, or UV energy, well, mostly UV energy, and it harnesses the sun's energy and helps you, maybe not you unless you're a tree, helps plants make glucose. So plants make their own food, which is glucose, with the chloroplasts via photosynthesis. And then with the glucose that they make, they can then use their mitochondria to break down the glucose that they made into ATP. So you eat things to get glucose, and then you break down glucose into ATP. Plants make glucose, and then they can use glucose to get ATP. Chloroplasts also have their own DNA. So it's also believed that chloroplasts also were bacteria that a cell ate once upon a time and then started photosynthesizing for them. So plants actually have 3 different sets of unique DNA: their mitochondria, their chloroplasts, and their nuclear DNA, their DNA that is in the nucleus of the plant. Plants also have a cell wall, so the yellow thing that says cell wall on it. The cell wall is made out of cellulose. Cellulose is a type of sugar. This cellulose is something that you cannot break down because you don't have the enzyme to. Rabbits do. So rabbits can eat grass and get sugar from it. You cannot get sugar from grass, so you can chew all of the grass you want, and it has no value to you other than giving you fiber. So the cell wall of plants to you is dietary fiber. So you've probably heard about this in commercials for like cereal and different things like that to give you fiber. Fiber is good for you. You need to eat fiber. That's why eating plants is a good thing. And it's not only plants that have fiber. Bacteria also have a cell wall. In this diagram, it is also yellow. The bacteria that have cell walls are very hard to kill. The outside of them is just different, and it's harder for you, your immune system, to find them and kill them, unfortunately. All right, so your centrosome is another organelle that's very important, and your centrosome is made out of centrioles, and there's 2 of them, so you can think of them as twins. So you have Bob and Juan. If you see just Bob on the street, you can call him Bob. If you see Bob and Juan, you can call them twins. So when the centrioles are together, you can call them a centrosome. When they are apart, you call them centrioles. So again, one more time, the centrosome, or twins, is when the centrioles are together, or when Bob and Juan are together. And when they're apart, you no longer call them a centrosome, you call them centrioles. So they each have a name, which is centrioles, and then together they also have a name, which is centrosome. The green thing in this diagram is your cytoskeleton, along with the red stuff. But anyways, your cytoskeleton is a whole bunch of proteins that hold everything together in your cell. So for instance, without your cytoskeleton, if you started jumping a lot, all of the organelles in your cells would fall to the bottom of your cell. That would be bad. You don't want that to happen. So it gives structure, it helps cells move. It's very, very, very important for the cell, just like your skeleton is important for your life, as an example. Okay, moving on. So, this is what you looked like 15 to 16 years ago. That is a fertilized egg. You got that way by the cell cycle. So the cell cycle is how you go from 1 cell to 2 cells. So, if we look here, we have interphase, which is the first phase of the cell cycle, and then the mitotic phase. Interphase is made up of G1, S, and G2. And then the mitotic phase is mitosis and cytokinesis. Cheers. All right. So in G1, it's the first step of interphase. This is when the cell grows. It starts getting ready to divide. You start duplicating things. It's the first gap. So G stands for gap. So it's the first initial time when your cell is getting ready and doing all of its things it needs to do. And then after the gap, in between G1 and S, you have a checkpoint. This checkpoint is responsible for checking your DNA to make sure everything in your DNA is good to go. So if you have any mistakes, your cell will try to fix them. If it cannot fix them, what will happen is your cell will kill itself. It's called apoptosis, A-P-O-P-T-O-S-I-S, apoptosis. And this is because if there's damage in your DNA, you do not want to make a copy of it. So you do not want to have two cells with damaged DNA. This is one of the ways that cancer can start, is when you have damaged DNA and the cell continues to grow even though it's damaged. So in S phase, once the cell has either fixed its mistakes, didn't have any mistakes, or technically it doesn't go into S phase if it dies, but moving on. So in S phase, the cell will duplicate its DNA. So you take your blueprint, your instruction manual for your cell, and you photocopy it. You make a second copy. After you make the second copy, before you go into G2 phase, you double check it. So you make sure that the copy is actually good. And again, if there's problems, you fix them. If they're too big, the cell will commit programmed cell death, or apoptosis. So it's like hitting the abort self-destruct button of your cell. In G2, the cell finishes growing, finishes everything it needs to do in order to go into mitosis. Now, before we go into mitosis and cytokinesis, or the mitotic phase, I'm going to teach you a little bit about your DNA. So, in the center of this diagram, you have your DNA helix. So what your DNA is, and then what you do to organize your DNA a little bit better is you take your DNA and you wrap it around histone proteins. Histone proteins are a way that you can just organize your DNA a little bit better so it's not as long, not as fragile, and just better organized. So when you have histone proteins and your DNA is wrapped around them and then that's wrapped around each other, you get chromatin. Chromatin is the spaghetti-looking thing in your nucleus. And that's what it looks like most of the time in your cell if it's not dividing. So, your chromatin, before your cell divides, has two copies of it. So you have two blueprints inside of that spaghetti factory mess thing. So if I was going to share a plate of spaghetti with a friend and just stabbed the fork in it and went to move half of it onto their plate, you wouldn't easily divide it into two equal halves. It just wouldn't happen. Usually the whole thing would fall onto the other plate. Therefore, you have to organize your chromatin a little bit better so that when you divide it into two, you get two equal parts. So what happens with your chromatin is it condenses even further into a chromosome, which is the thing on the right that says condensed chromosome. So this is what you normally see DNA as in like crime shows and stuff, like the different chromosomes. It has a centromere, which is just the center part of it, and then the arms and then the absolute ends are called telomeres. But now that we know what chromatin is, and now that we know what chromosomes are, we can actually go into mitosis. So this is the same diagram that we had earlier, but this one has your mitosis section expanded. So instead of just saying mitosis and cytokinesis in the mitotic phase, you have prophase, prometaphase, metaphase, anaphase, and telophase. What are all of those things? Lovely question. Here they are. So, In the first cell, we have G2 of interphase. G2 of interphase means that it hasn't gone into mitosis yet. So you still have chromatin in the nucleus, your centrosome is still together, and your nucleus is still there. So as I said earlier, in this unit, your DNA will never leave your nucleus, but your nucleus sometimes leaves your DNA. This is what I'm talking about. So the next cell over to the right is prophase. So in prophase, 4 things happen. Your chromatin condenses into chromosomes. Your nucleus starts dissolving. Your centriole, so now it's moving apart, so you just call it your centriole, starts moving to the other side of your nucleus, and it starts growing spindle fibers. And I'm sure that is written somewhere. Yeah. Spindle fiber. It's right there. Anyways, so spindle fibers start growing, your centriole starts moving to the other side of your nucleus starts dissolving, and your chromatin starts condensing into chromosomes. Now, when I was a student your age, my textbooks went straight from prophase to metaphase. However, that was kind of confusing, so they've added in prometaphase, which is really good because it explains how you actually get to metaphase. So, in prophase, all of those things are starting. In prometaphase, all of those things are finishing. So your nucleus is finishing dissolving. Your chromosomes are finishing to condense. Your centriole has moved to the other side of the nucleus, and your spindle fibers are finishing growing. Now, the other thing that happens is your spindle fibers attach to the kinetochore, which is just the attachment point of the spindle fibers to the center part of your chromosomes. So your kinetochore is just kind of like the handle for the spindle fibers, so that when you break your chromosomes in two, that's how they are attached to the spindle fibers. Then, once all of that happens, we can now eventually get metaphase. Metaphase is not much of a process like the other two that we just talked about. It's when something has finished happening. So you're only in metaphase when all of your chromosomes are lined up along like the equator or the center of your cell. This equator is known as your metaphase plate. So the metaphase plate is basically the center part of the cell. So all of your chromosomes are lined up in the middle of the cell. After metaphase, we move on to anaphase, which is when your chromosomes actually split into two. Your chromosomes actually split into two so that they start migrating or moving to the other sides of the cell. So when the cell splits, you get one set of DNA on one side and one set of DNA on the other. And when the chromosomes split, or when your cell eventually splits, you refer to it as your daughter chromosomes, or your daughter cells. So when cells split, it's not like mother and kid or whatever, it's daughter. So then after anaphase, you get telophase. This diagram says telophase and cytokinesis, but cytokinesis has not happened yet, so just ignore that part. So telophase is kind of like the opposite of prophase. So in telophase, your nucleus starts forming, your chromosomes start unraveling, so the opposite of condensing, back into your chromatin, so you get your spaghetti back, and then your spindle fibers dissolve, so they go away, and then you make something called a cleavage furrow, and that's kind of like the pinching off of the cell, so it kind of looks like a snowman now. So cleavage is when you split something, and the furrows, you can think of it like a gap or a ditch or a line. So the cleavage furrow is where the cell will eventually split into two, and once it splits into two, that is known as cytokinesis. But until then, it's in telophase. So if you have, once it splits, that's cytokinesis. Before that, it's telophase. So now if we move on, just a different diagram, and again, all these diagrams you can find just by Googling the cell cycle on Google Images. So again, it's the same cycles, same phases of mitosis, but these ones are paired with microscope slides. So the first one, you can kind of start seeing your chromosomes in prophase when they start condensing. Your chromatin condenses to chromosomes. And then the next one, you can definitely start seeing the pink of where the centrioles are. Your chromosomes are definitely visible and everything's starting to organize. And then in metaphase, you can see they're all lined up in a line. In anaphase, they're split and they're moving to opposite sides. And then telophase, you can see the cleavage furrow as well as you can't really see your chromosomes anymore. You can't see your chromosomes anymore because they're unraveling, they're going back to chromatin, and you cannot see chromatin under a light microscope. So the next picture is, I believe, fake. This is not what it looks like. It just looks cool, so I hope you like it. So once it's actually split, once it's moved into two, then you will have two daughter cells, and that is cytokinesis. That's when you actually split the cytoplasm. And that is how you got to this. So that's when you were 6 cells large. And then that continued and continued and continued and continued and continued until you're several trillion cells now. So each time your cell divides, it goes through all of that, except for your sperm cells and your egg cells, which is not possible to have it in the same person. But anyways, guys, sperm cells, female, egg cells. Because they undergo something a little bit different, so that when you do make an egg cell, you have only one full set of DNA instead of doubles. So if you have any questions about that, please come and see me. This is so that, again, Thursday and Friday sections, you will have all the information you need for the quiz. So please watch this sooner, so if you have questions, you can come in on Friday, because you will need to know all of this for future quizzes, as well as some other things. So I hope that helps. If you have questions, let me know.